Section 3.3 talks about the cell envelope, which is going to be comprised of 2 to 3 basic layers. It's going to be the cell wall, the cell membrane, and in some cases an outer membrane. But all of these components comprise the cell envelope, and that lies outside of the cytoplasm. First, we're going to talk about the peptidoglycan cell wall. This is going to be a major part of bacteria cells because almost all bacterial cells have a cell wall. What peptidoglycan is a framework. You can kind of think of it as a scaffolding around the exterior of the bacteria, and you can think about it as scaffolding around a building that's being built or remodeled on the outside. And it's a framework of these chains that are cross-linked by peptide fragments or protein fragments. So again, they're present in most bacteria. And the purpose really is to provide strength and to provide resistance to the cell rupturing due to osmotic pressure. And here's a very basic diagram of the repeating framework of the cell wall. So you have alternating links of this G and M. All right, the G stands for N-acetylglucosamine, while the M stands for N-acetylmuramic acid. All right, these are going to be the repeating framework, and then they're going to be cross-linked, you see here with the yellow bars, with the proteins. This is a picture from a different textbook, and I just like it just a little bit better. You've got the sugar backbone here of the G and the M repeating subunits, but then there are 2 types of cross-linking components. All right, the tetrapeptide amino acid side chain in the orange or brown, tan, whatever this color is, and then the pentaglycine amino acid cross bridge, where you're going to have 5 amino acids. And this just creates a very tight network of structural components that gives structure to the bacterial cell wall. Now, many years ago, very long time ago, a scientist by the name of Hans Christian Gram developed what he called the Gram stain. And by doing the Gram stain, he was able to differentiate between 2 major groups of bacteria cells. The first is going to be the gram-positive cell. The gram-positive cell has a very thick layer of peptidoglycan, as you can see here. The peptidoglycan is going to grab onto the first stain within the Gram stain process. That's going to be from crystal violet. That's going to cause the cell to appear purple. All right, that's why this color, or the peptidoglycan here, is purple. All right, also specific to gram-positive cells, not identified 100 years ago or more than 100 years ago with Hans Christian Gram, but later on when technology became better, it was the identification of these different teichoic acid and lipoteichoic acids that move through or are embedded directly into the peptidoglycan layer. And then also characteristic of gram-positive cells is just a single cell membrane here. And as you can see, that is composed of those phospholipids. So it's a phospholipid bilayer because there's 2 layers of those phosphates. All right, then a gram-negative cell looks a little more complex than the gram-positive cell. All right, so here what you have is a very thin layer of peptidoglycan. And if you compare this layer, where you see just 1 layer of balls depicting that, in here, you've got the very thin layer of the peptidoglycan. Just 3 or 4 rows of these ball-like depiction of the peptidoglycan, whereas here you have this very thick layer. and maybe there's 20 or 30 of these different layers of the peptidoglycan. So a big difference. All right, this one is not that dark purple, but rather it's more of a pink or a red color. And that's because when a cell is stained with crystal violet, right, that very thick layer is stained, or both layers initially are stained with the crystal violet. The second step in that Gram staining process is the iodine, which acts as a mordant to really lock that crystal violet color in. The third major step of the Gram stain is going to be then to decolorize the cell with alcohol. And in both cases, some of that crystal violet comes out of the peptidoglycan. All right, the thin layer of peptidoglycan with the gram-negative cell. That alcohol washes out all of the crystal violet. Whereas with the gram-positive cell, maybe that very top layer of the crystal violet is drawn out by the alcohol, but there's still a lot of that peptidoglycan layer that still maintains that crystal violet. Then the last major step of the crystal violet, or the Gram stain, is to stain it with a secondary stain called safranin that colors the peptidoglycan that pink or red color, and that's what you see here. The gram-positive cell might pull in a little bit of the safranin stain at the surface, where a little bit of the crystal violet may have been washed out, but that crystal violet is so dark and so potent that as long as there's a little bit left, what you see on the microscope is always going to be that purple. All right. Now, the other major component of a gram-negative cell is going to be lipopolysaccharides, also called L for lipo, P for poly, and S for saccharides, LPS. This is a major component within the gram-negative cell that is specific to the gram-negative cell. All right, then another difference between the two types of cells is that with the gram-negative cell you have two membranes. You have an outer membrane and then an inner membrane, or what's called the cell membrane. All right, and then that has the peptidoglycan in between the two. All right, there's also what are called porins. All right, those porin proteins are going to create these pores that allow material to move through that outer membrane versus just moving directly through the peptidoglycan, like it would in the gram-positive cell. So the gram-positive is a way to very broadly differentiate the types of bacteria cells. Another way to classify cells is going to be acid-fast. Right? And acid-fast bacteria typically do not stain well with the gram stain, and so therefore it's hard to identify them, whether they're gram-positive or gram-negative. So most bacteria are going to be non-acid-fast. And this stain, well, let's go through the bacteria. There aren't very many acid-fast bacteria species out there, or a genus. The Mycobacterium are acid-fast, as well as a species, or the genus Nocardia. Mycobacterium is the pathogen that causes tuberculosis, as well as leprosy. And doing this acid-fast stain is very important to identify these two organisms. So, what makes these cells acid-fast is a component called mycolic acid, and it basically is a wax. And wax is just that, it's wax. It's kind of hard, and when doing the gram stain, it's very difficult to get that color or get that stain into the bacteria cell wall because it's wax. So what the acid-fast stain does is it utilizes heat, and you put a stain on the cell while you're heating it, and you use that heat to basically relax that wax, not necessarily melt it, but relax it, and so it will draw the stain in. You do a a decolorizer stain, but once that mycolic acid cools down after it's been heated, nothing's going to wipe or decolorize that initial stain. And so if a cell is acid fast, then it's always going to show up that color that was used while heat was being applied to the cell. So very important to identify these organisms. Now another group of bacteria cells, which we're really not going to talk about at all, are those that are deficient in their cell wall. So while most bacteria cells do have a cell wall, there are some that are going to be deficient. And there's a couple different kinds and different ways that it's thought that these cells will develop or have developed, but they're not very common. So that's why we're not going to do anything more with that. All right, moving back to LPS, the lipopolysaccharide. This is again located in the outermost layer of the outer membrane in a gram-negative cell. And some different names for it are going to be lipid A and then endotoxin. And the lipid A is just part of that molecule. If you remember from the picture just a few slides ago, it was just, like a half an inch relative to the size of that cell of a piece that was sticking out of the cell wall. Right? There's a couple of different components of that molecule, but one major part is this lipid A component. The lipid A is also called endotoxin, and this stimulates fever, and in high amounts it can also stimulate shock. So if you have a bacterial infection and you are running a very high fever, and especially if it's something on the interior, it's stomach related, it's GI related, all right, the physician's going to wonder or think that you have a gram-negative or an infection being caused by a gram-negative organism, and therefore are going to give you an antibiotic against gram-negative bacteria and not gram-positive. All right, then the last bit of information regarding the cell envelope is the cytoplasmic membrane. All right, the cytoplasmic membrane is the lipid bilayer. All right, so that lipid bilayer with proteins embedded. Bacterial cell membranes contain primarily phospholipids as well as proteins. So 30 to 40% of the membrane mass are these phospholipids. Then 60 to 70% are going to be different proteins. Two important features or functions of the cytoplasmic membrane. Right? Prokaryotic cells do not have mitochondria. In the eukaryotic cell, mitochondria are going to be the location for the process of the electron transport chain, right, where cells are going to harvest energy, create and harvest energy in the form of ATP. Because bacteria lack mitochondria, the cytoplasmic membrane is going to be where those reactions are going to occur. So ATP is generated within the cytoplasmic membrane in bacteria cells. And I believe we'll talk about that process in more detail. It's either chapter 6 or chapter 7. Chapter 7, where we'll talk a lot more about that. All right, the other basic feature of the cytoplasmic membrane is that it regulates the passage of nutrients into and out of the cell. And that's it for section 3.3.